Many people conduct research that are not valuable, that cannot see the light of the, that cannot go beyond their laboratory, that cannot go beyond their university. The research you do should be intercontinental, should be some a research that anywhere in the world you can be invited to talk. And this is how it starts. And that is why I'm putting your own voice is out there. Your own voice is out there on the research space. There's a professor in Nigeria here in the University of Ilorin where I lecture, is in the Faculty of Agriculture, Animal Production. He said he was just publishing his work and he was contacted from abroad to come and speak on that particular thing he's doing research on. And they paid for his transport, his tea fare, his plane, everything. They paid for his hotel abroad. They gave him money also. And they took care of him and interacted with them and he came back. So at a point, people want to see who is the person. They want to meet with you. Despite that you are talking like this in the media, they are seeing your voice. They also want to see you face to face. They want to learn from you. And I have seen that international collaboration is very, very important. And that is what is because um, I was working on a research some time ago and I happened to win the Fulbright to go to USA. And the first week, I had to make presentation of what I've done. They are interested as much as we think that we don't have enough equipment. They have seen what you are trying to do. They want to know what is it you are trying to do. They want to tap into your idea. So that is part of it. So if what you are pushing forward is not, even with your small means, is not valuable, it's not scientifically based, the other people will not invite you. The other people will not take you serious, except in some other way. So I want us. That's why these are the things that make microbiology research valuable, and that is what we are going to be looking at today. So the first thing we are going to talk about today is building collaboration and networking for impactful microbiological research. It is important that we build collaboration, and if you don't collaborate, I don't know how or what type of research you are doing. So I'm going to start with the first one in earnest, building collaboration and networking for impactful microbiology research. Collaboration and networking are essential components of impactful microbiological research. In today's rapidly evolving scientific landscape, interdisciplinary approaches, shared resources, and collaborative efforts are crucial for advancing discoveries, securing funding, and translating research into real world applications. I have friends in chemistry, I have friends in physics, who are friends in plant biology, our friends in agriculture, they design their research and then the microbiology aspect, they push it to me so that I can be involved in the research. I also write researches and I put some chemical components into it and I involve those in chemistry. No individual can do all the research alone. Even within microbiology, even within biotechnology, you don't just hold on to people in one area alone. You look for people in some area so that they can bring in that expertise into the work. Now, talking about collaboration, I was working on some chemical compounds and looking at how microorganisms can be used to degrade them. And during the presentation, the structures we made, they were not of equal sizes. Somebody who was in chemistry was in the meeting during the presentation and he said in chemistry they have to be in equal sizes, all the structures. When you say arcane, when you draw pentane or hexane or cyclohexane or benzene, the structures must be of equal sizes. So you can't draw benzene very big, then you draw arcane, you use it very small, even though you get the structural formula, even though you get everything you need to get, but the shape or the size must be of equality. So you can see what that contributes if you are going to publish in a chemical journal, in a chemistry journal. It's going to help your work to go far. Compare with if you just stand as a microbiologist or as a biotechnologist without involving experts in the different fields you are working on, the work is going to have some kind of difficulty to scale through. So this webinar explores the importance of collaboration in microbiology research, strategies for building strong professional networks, and ways to foster effective partnership for groundbreaking discoveries. Now, why does collaboration matter in microbiology research or in biotechnological research or in any other kind of research? Microbiology is a vast field that intersects with medicine, biotechnology, environmental science, and public health. So microbiology intersects with many different fields outside of even what I've mentioned. So collaboration now will allow researchers to access diverse expertise. I've explained this already. So you work with specialists in genomics, bioinformatics, immunology, and epidemiology. It strengthens research outcomes. I have a collaborator with a one university in India, still in the, I think, in one of the universities, one of the state university in Surat. And then I mean, with in touch with the HOD there, they focus more on biotechnology. So when there's collaboration, now the genomic part is like they are very good. They focus on that part. They focus on bioinformatics. They focus on all those couple of parts. So those kind of people now, any work that relate to bioinformatics, you push it to them. While the other people do the other area of the work. So that will make you to access diverse expertise. Then, apart from this, collaboration enhance research funding opportunities. If you are looking for research funding and you are just narrow in your research, it's going to be difficult. But when the research is interdisciplinary, you see that you get a headway. So funding agencies favor interdisciplinary and multi-institutional research. So multi-institutional, you can see, not just from one university. This one is from the Polytechnic, this one is from the College of Education, this one from university in Nigeria, this one is from university in the north, this one is from university in the south, university in the east, university in Asia, university in India, university in different places. It shows a lot to the funders. So it also improved data collection and analysis. Shared laboratory techniques, equipment, and computational tools lead to more robust and reliable findings. Where one person is weak, the other individual in the research is strong. Like I am very, very strong in writing. I'm very, very strong in some laboratory aspects. Somebody else may not be strong in writing, but the person may be strong in some other aspects. Maybe in applying for funding. Applying for funding is another field, is another area on its own. Somebody can sit down and make sure you apply for funding at least once a week. And that is a big one for any research group, and it's applied on behalf of the group. When the grants come, the group, all the members of the group are going to take part in the grant. So that shows you. So it improves many things. It also facilitates faster scientific advancements. Research breakthroughs often come from combining ideas across disciplines. So now, faster scientific advancement. When you are in a research group, you don't need to you are not solo, you are not just alone. So a paper is you are working on a paper in the group or two papers in a year, maybe literature review, maybe book chapter, you are working on this, you are working on that. So everybody now, at least from that research group, you are sure of two or three papers or two or three book chapters in a year, and that is a big one for the research group. Now, let's look at the strategies for building strong professional networks. To establish successful collaborations, microbiologists must actively engage in networking opportunities. And here is how you can do this. Number one, join professional societies and research groups. Becoming a member of organizations like the Society for Experimental Biology of Nigeria, which I am the General Secretary in Nigeria here. You see, when I write my CV anywhere and I put them general secretary, in fact, they don't look at it. They know that this person, for him to be a general secretary of this society, not just a member now. I'm a general secretary. Previously, I was the assistant secretary. Now I'm the general secretary of the society or the whole country. That shows is a big deal. And the who is who in the experimental biology field, I know them and they know me. So now, when they want to review for professorship, review for grant, review for many things, the person they will send it to is somebody that know me. Maybe even I'm even the one that already started them as members of the society. Maybe I'm even the one that gave them some information at some point. So they know me already, and that helps to move you forward. Then also we have the American Society for Microbiology, which I'm also a member. Then we have the Microbiology Society in UK. I'm also a member of that. Then we have the Federation of European Microbiology Societies. So make sure you are a member of a professional society that you are interested in the scientific field. Don't just stand by and just be a member. Be a member, be active, attend their conferences and continue, attend their AGM, contribute, do something. In fact, aspire to be one of the ESCO and even be for a little while and then you leave. Not that I'm not saying you should go there and be there forever. Be part of the ESCO, take this role, take that role, take this responsibility, and take that responsibility. And that, in fact, that is how you can now help your research group. There are some things you will learn in that space. You can use it to help your research group. There are some things you will need in that particular society. Your research group will help you, and that will build your professional capacity. That will help to build your CV, build your capacity in your university or in anywhere you are doing your research. So these platforms offer access to conferences, research funding, and networking events that connect microbiologists with potential collaborators. So this research group we are doing now is just, it looks small, but it's a big deal. Many societies that we are talking about, they don't even do up to what we are doing. All they do is hold conferences and they move on. And then even this research group, we can take it up to the next level and register it, and it becomes a big deal. But for now, we are focusing on building our own capacity and building our research capacity and other capacities. Another thing about the research group is for you to be able to take part in it, or how you can build strong professional network is that number two, attend conferences and workshops. While I was in the US, I attended 3 conferences within 9 months. I attended 3 conferences within 9 months. I was in the US. I got to the US in August 2015 and I left in May 2016. I attended one conference in February, another one in March, another one in April. So I attended 3 conferences within 9 months. And in all, during the time of my PhD, I attended 6 conferences. I attended 6 conferences within the time of my PhD. So all these, I put it in my thesis, in my PhD write-up. I put it there at in the appendix, conferences attended, and I put the list. Even the external examiner we know that this person is coming to examine his stuff. And I published 2 papers from my PhD thesis before the defense, and I put them there in the appendix. I listed them, and I also put the papers at the end. One of the questions they will ask the examiner, which you don't know before they examine you for PhD, is that this work you want to examine, this PhD that you want to come and do the viva for, is it a work that can be published? Is it publishable? That's part of the question. Now you are already published the work, part of the work, and they are asking the man, is it publishable? And he sees the appendix that you have published the work. What is he going to say now? You are published already, said, so you have helped to make the work of the external examiner easy. Immediately we write it there that yes, this work is publishable. And sometimes even where you publish, the external examiner does not know about that place you published, and then he's coming to ask you, where did you publish? And he's asking you where you published, and he also want to write down the name of that journal, and you want to publish there. So don't see yourself as a small fry. You are a big deal if you know what you are doing in the research group and in your academics. So you attend conferences and workshops. Scientific conferences provide opportunities to present research and receive feedback from experts. So you present your research and you receive feedbacks. It's very, very vital. When I was in the US, the first presentation I made, everybody was saying very good, very good. My supervisor said no, it's not very good because it was seated. Then somebody raised the question that I work with only alkane, only hexadecane, and I'm generalizing for alkane. And based on that comment, we went back to the lab and we looked for hexane and cyclohexane, because that means somebody can be thinking in that direction. That's a feedback for us. Then you also meet potential collaborators for multidisciplinary, multi-institutional studies during conferences. Then you learn about emerging technologies and innovative research methodologies. So another thing you need to do is to utilize online networking and collaboration platform. Researchers can connect globally using ResearchGate, and I use it a lot. I met a collaborator on ResearchGate, and we have published 2 papers. Funny thing, we have not met face to face, and we have published 2 papers. He helped me with submitting my sequenced data to the gene bank, NCBI. He helped to submit them. I didn't know about those things. I got them when I was in the US, did everything, but I didn't know I was supposed to submit them to the gene bank because they are new isolates. And when you look at it, he told me, and he submitted it for me. When I wanted to make correction on it, because of the name, he didn't put the name of my supervisors. And one of my supervisors saw it and said his name is not there. So I had to go back, and he told me how to do it, and I did it myself. And I changed the name and I put all their names in it. So if you go to the gene bank, NCBI gene bank now, put my name Adetitun on the gene bank, you will see the work I submitted to the gene bank with the sequences, everything will be out. So apart from ResearchGate, you also have Google Scholar. and ORCID to track research impact and citation. Then we have LinkedIn and Twitter to engage with microbiologists and industry professionals. See, all the social media that people are using to for rubbish, you can use it to advance yourself, like YouTube. Like now we are using Google Meet. Some people are using Google Meet for nonsense. Some people are using Twitter for to post nonsense things and to just go there and just look at gossip. There are also microbiology content there. There are also scholarship opportunities there. There are funding opportunities there. There are so many book writing opportunities there. People are inviting for book chapters. You can find them there. And if you are sure that they are legitimate and they are real people, then you can go in and work with them. Now, let's look at how to build effective research collaborations. I hope you are following me. If you are following me, say we are following. Hello. If you are following, can you? Okay. Amina, are you following? Oyetayo, are you following? Ajadi, are you following? Can I hear your comments? Ajadi, Oyetayo, Amina. Can you comment? You are following?
I'm following, sir. I'm following, sir. Yes, sir.
Okay, Ajadi, what about you? Are you with us? Ajadi Olanrewaju. So, not everybody is with us. Some people just set something up and they go. And it's going to show later on. But we at our end will do what we can do. And just like in the class, some will be talking, some will be paying attention, some will not be paying attention. So this opportunity that we have, we should utilize it properly. So I said on LinkedIn and on Twitter and on all social media platforms, people use it for different things, but we can use it to assess and to promote and to engage and to make our research visible, make ourselves visible, make your research visible. Somebody is saying that in the era that we are today, we are in the information era. Now you have information. Now you have knowledge. You don't let people to know you are existing. How people will not know you are there? You are just in the corner. Only people, your local people know you are there. But when you project yourself on social media, you show yourself. There are different social media platforms. You go there, answer microbiology questions, even on ResearchGate. Go there, register your account, then answer microbiology questions. All your papers, put them up on ResearchGate. Create your good profile for yourself on ResearchGate. People will contact you. You'll be surprised. They will contact you. Wow, you are working on this, you are doing this, and people have contacted me, and I have also contacted many people on ResearchGate. So now let's look at how to build effective research collaborations. Once networking efforts result in potential partnership, it is important to establish clear collaboration strategies. So the first thing you have to do is define clear research goals. Before starting a collaboration, discuss research objectives and specific contributions of each partner, expected outcomes and timelines for the project. Most times in the local setting, people look for their friends to do research with. They look for only their friends. Now, don't do research with only your friends. Look for people that you see that these people, they know what they are doing. They know this thing, and then you can learn from them. Yes, do research with your friends, but definitely your friends will be people that are also serious like you. But don't just limit it to your circle of friends. Look at social media and look for people that you think they can contribute something valuable to your research you're doing. And that is what you should do. So we also have number two thing here is leverage shared resources and expertise. Exchange laboratory techniques, e.g., CRISPR, gene editing, next gene sequencing, bioinformatic tools. So you can exchange laboratory techniques like that. Then collaborate on sample collection and field studies across different locations. You see, you can arrange a research whereby you are in Nigeria, somebody else is in India, somebody else is in USA. So you want to work on river sample. You want to sample from a river. Now you are sampling from a river in 3 continents, or you can sample from rivers in all the continents. So you pick one river in Ilorin, Nigeria, pick one river in Surat or Badoli, India, pick one river in Oklahoma, USA, pick one river in UK, pick one river in Australia, pick one river in another place, and you have collaborators there. So everybody is working on the same thing. This is what you are doing, this is what they are doing, this is what they are doing, but different rivers. Now all the results, you bring it together. Then you write up the paper together. This is what we found in this river, this is what we found in this river, this is what we found in this river, this is what we found in this river. Instead of you just writing on only your own local river, you have now written on all rivers across the continent. And you have collaborators publishing together across the continent. That kind of work is going to be well noted, it's going to be full, it's going to be loaded. So you use shared data repositories to enhance reproducibility and transparency. Sometimes I post papers that we want to read on the group. Now some people ask me that they can't access the paper. Why can you say you're a researcher and they give you a paper and you say you can't access? Some will say you have to pay before you access the paper. All you need to do is to look for somebody in UK, look for somebody in the US university who is a collaborator. It can be your friend that travel, it can be somebody that is there that you have met online. Send the book, the chapter of the paper to them, or the name of the paper and the DOI number. The person will use the free opportunity to download that paper in their own institution and send the PDF to you. So that was what I used to do. I would send it to like 3 or 4 people, and most times 3 or 4 of them will reply me with the paper, though at different point in time. So this is how you work. This is how you do your things. Don't just stay and you just say everything is just within. You send far away and you interact with people outside of where you are. Now you also have to secure joint research grants and funding. And that is where we'll be taking our next line of action to. We have to look for a research grant we are going to write jointly. So we secured the research grant. While I was in India, I wrote a series of research grants where all of them replied negative. But that is not a big deal. I've learned a lot from that. And I have many research grants that I'm writing, many proposals I'm sending out. Most of them are returning negative. But I tell some people, if only one just return positive, things will change things. It will change things. I met a woman at the conference from USA, from SOT, Society for Toxicology, and we were talking about grants. And I asked her, she said she writes 12 grants a year, and she wins 2 out of those 12. And those 2 can carry her for 6 to 7 years. So she still have a grant that's carrying her for 6 to 7 years, and then she's still writing 12 grants. And she wins 2 again, though that's 2 will carry her again for some time. So scientists should not depend on their salary alone. You should be secure research grants and funding to fund the work you are doing, and that is going to help you to move ahead, because inside of the funding there's going to be money for the principal investigator and for the other investigators that are going to do the work. So in some places people are using their money for research. We should be thinking of writing research grants, applying for scholarship, applying for funding, and that is the way to go. So funding agencies like National Institute of Health, the World Health Organization, and the Bill and Melinda Gates Foundation support collaborative microbiology. Researchers can apply for multi-institutional grants to increase the success rate of funding proposals. Now, we also need to maintain effective communication. Successful collaborations require regular virtual or in-person meetings to discuss research. Can you see that what we are doing now? We have regular virtual or in-person meetings to discuss research. Regularly, we are meeting virtually like this. We are discussing our research. Then use of project management tools like Slack, Trello, or Asana to track tasks. Then resolving conflicts professionally to ensure smooth research execution. Now, let's look at examples of successful microbiology collaborators or collaborations. The Human Microbiome Project, HMP, is a global effort to study microbial communities in the human body, involving researchers from multiple disciplines. The COVID-19 Vaccine Development Consortium, collaboration between microbiologists, biologists, and pharmaceutical companies to develop vaccines. Then we also have antimicrobial resistance research networks. And let me tell you, somebody came from the National Universities Commission in Nigeria to my department in December for NUC accreditation. They do accreditation in Nigerian university every 5 years for each program. So if you get full accreditation, the another 5 years you have to do another one so that they can see your facilities, see everything, see your staff, see everything you have. So the man asked me, which field am I working on? And I told him environmental microbiology, basically biodegradation of petroleum contamination or thereabout. And he said, why not AMR? He said antimicrobial resistance, there's money in it. Money is being pumped into that kind of research. So what do you do then? What I do most times is that when I isolate juice organisms for biodegradation, I also use them to check for antimicrobial resistance or antimicrobial susceptibility or antimicrobial inhibition to see, okay, this organism that can degrade oil, what else can it do? And that is what I learned when I went to the US. Before I went to the US, I was working with so many organisms. When I got to the US, Professor Babu Fatepure, he told me, he said, what are we going to do with many organisms, more than 20 something organisms? So you have to narrow them down to 4. In fact, he will have wished it was only one organism that will narrow down to. So that just that one organism, you can do different tests with it. See its ability to do this, ability to do this. And that was why in the US, I checked ability for it to degrade, for the four organisms to degrade benzene, to degrade toluene, to degrade ethyl benzene, to degrade xylene. I tested for ability to degrade hexadecane, to degrade hexane, cyclohexane, benzoate, and then phenol. You can see because there are four. But assuming that it's just the 20 something, it will be too cumbersome. So you have to look at ways to narrow down your research. As a conclusion, I need to say that building strong collaborations and networks in microbiology research enhances innovation. It enhances funding opportunities and scientific impact. By leveraging interdisciplinary research, sharing resources, and maintaining strong professional relationships, microbiologists can drive groundbreaking discoveries that address global challenges such as infectious diseases, antibiotic resistance, and environmental biodegradation and environmental sustainability. So, what I'm trying to drive at is that if you are going to collaborate, that means you have some capacity. You cannot collaborate on empty, just on empty thing. You have to have capacity to collaborate. So your capacity to collaborate is what will help you in your collaboration. Your research collaborators want to see what you can do. Some people you give them tasks to do the way they are doing, the way they are doing. Some will even give excuses. They will give so many things. They do so many things. If you are like that, your research collaborators will do away with you in a short while. They will note you and do away with you if they themselves are serious. So if they give you a deadline, you meet the deadline, you beat the deadline. You give a deadline of 7 days, in 4 days, you already delivered. So the person know that if I need something urgently, this is the person I should call to. If I need something to go out urgently, I can call on this individual and is going to do it. If I need something tangible to be done, this is the person I should call to do it. So build your capacity. That is how you can be valuable. So when you build your capacity, so in NS, collaboration and networking helps you as a researcher, it helps your research to be valuable. If you don't collaborate, your research is just you. Just you. Everything you do in your research is just only you. You don't collaborate. You didn't talk to anybody, any other person. Only you just doing your research. Car. I don't know what that kind of research is going to be turned to. You have to ask other experts to give input. You will learn from them in the process. So you are learning in the process so that by the time you finish, and that is why I removed some people from the group. They have already finished their PhD. And they were not under me during their PhD. They finished already. Now they want to be in the research group. No, you should have learned what you're supposed to learn. Then after you have finished, you can go and form your own research group. Since I was not your trainer. So they should go and form their research group. People that finish, they should not join my research group. And that's why I removed some people from the research group. But if you have opportunities, you can invite them once in a while. But that is basically why I remove them. And I've told us at the start that I want us to build capacity. That's why we are doing all our webinars in-house. I have people ready to talk to us from outside. If they come to talk to us, you are building them. So when you are giving the money to talk in the research group like this, you yourself, you are building your own capacity. You can go back to your own video and go and listen. I listen to my own videos. I do a lot of videos. I do a lot of recordings of my own speeches. Any speech I give anywhere, I record it. And I listen to them again. I'm wondering, so I'm the one that talked, that said all these things. Yes, that is it. You build your capacity. You don't know what you can do. You don't know what you can achieve. You don't know what you can actualize, except you keep trying. You keep making effort. If I now, members of this research group now, I can bet it on anything that if you are invited to come and give a speech somewhere, it should not be a problem for you. Just that some people will speak for 5 minutes, some people will speak for less than 10 minutes. When you are giving 10 minutes, you are speaking for 5 minutes, speaking for 3 minutes. That is where you should build up on. Get material, know how to space and to pace things, and get materials that will help you to be able to scale up and be able to do what you're supposed to do. Let's assume that I'm invited to speak somewhere and I cannot go. I can look at people and say, okay, you go and speak in that place. Or somebody else is invited, they can say, okay, please, I have an invitation. Can somebody else speak on my behalf at that event? That is what you should aim at, number one, from research group, in addition to others. So when you are given opportunity to speak like this, give it your best. Give it your very best. So this is what I have to tell us about building collaborations and networking for impactful microbiological research. I don't know if you have any question before we go to the next one. Do you have any question before we go to the next one? Any question? Any question before we go to the next one? Okay, in the absence of any question,
Thank you. Thank you, sir.
all right,
I don't have any questions, sir.
all right. Do you have anybody with question before we go to the next one? All right, we'll move